Hi everyone, welcome back. So what we just did was we went into this gross anatomy, this view from the naked eye. We talked about muscles and their different components, and then we went into the structure of gross anatomy. We separated these muscles versus fascicles versus myofibers, and we looked at the different connective tissues surrounding each of those. So now what we want to look at is we're going to look at this individual muscle cell, this individual myofiber. That's this guy right here. Individual muscle cell, and we're going to see that it's made up of these different things called myofibrils that have these things called myofilaments, and we'll see some sliding filament. All of that stuff is what we're getting into now. So I'm going to write up a few different words that we want to become familiar with before we start talking about these muscles. Alright, so as we're getting into this microscopic anatomy of the skeletal muscle, when we're looking at cells, we're typically looking up under a microscope. So what cells are we looking at? We're looking at these muscle cells, these myo meaning muscle fibers. So these are single muscle cells. Whenever we learned about tissues, we talked about the four classes of tissues: epithelial, connective, muscle, and nervous. Very briefly, we mentioned when we're talking about muscles, we know muscles have this modified plasma membrane called a sarcolemma. So that's going to be our plasma membrane of the muscle cell. And then we also know that we have a modified cytoplasm, and that's called the sarcoplasm. So modified cytoplasm. And what are we going to find within this cytoplasm? If you remember in Unit 1 we talked about some cytoplasmic inclusions or some cellular inclusions. So these do have some cellular inclusions, and the skeletal muscles do have some cellular inclusions. One of those is myoglobin. So you've heard of hemoglobin before. That's that protein that carries oxygen around in the blood. Well, now we have a protein in the muscle cell that also holds oxygen. So muscles need oxygen and sugar. You need the sugar to get ATP. We'll talk about all that later on, but we need sugar and we need oxygen. So how do we store sugar? We have these glycogen. We store glycogen. So glycogen is it's a polymer made up of those monomers glucose. So this is our sugar, and we break this down to get ATP. And these two things, we'll call these cellular inclusions, cytoplasmic inclusions, sarcoplasmic inclusions, whatever you want to say. Alright, so that's our sarcoplasm. We know nuclei whenever we did the classifications of muscle tissue. We know if we're looking at skeletal muscle, we know that these are multinucleated because the smaller skeletal muscles fuse during development. So we'll put multinucleate. And that's because the fusion of cells during development. Okay, and then if you guys remember, we talked about this thing making up the cytoplasm. The cytoplasm had different things in it. One of it was that liquid cytosol, but then we also talked about this thing, the cytoskeleton. So the cytoskeleton are these different filaments. So if you remember words like microtubules and intermediate filaments and what was the other one? Microtubules, microfilaments, intermediate filaments. Anyways, we're going to get some modification of those here, and that's going to give us our myofibrils. So these myofibrils are these modifications of the myofibril. Modified cytoskeleton. And the myofibril is the actual thing that's going to be doing our contracting. This myofibril can then be broken up into smaller things called sarcomeres, which we'll get into in a little bit. I'm going to leave this as myofibril for now. But when we look at the picture I drew back here, hold on. When we were looking at this, we know that each skeletal muscle cell has hundreds or thousands of myofibrils. So this is one myofibril. So if I was really drawing a single skeletal muscle cell, remember they're nice and long. So if I was drawing this like this, I would have this would be one myofibril. That would be a myofibril. That would be a myofibril. And then they're broken up into these smaller units called sarcomeres. And we'll look at the anatomy of a sarcomere in a little bit. But just so that you can see, this, I know I drew one back here. This was one myofibril in this skeletal muscle, but this skeletal muscle has hundreds of myofibrils within it. So myofiber has myofibrils. Okay, let's get rid of all this and keep up with some more anatomy terms. So we had myofibril. The next thing that we want to talk about is the sarcoplasmic reticulum. So this is another modification of an organelle that we've already seen. I'll let you take a guess while I'm writing it. So sarcoplasmic reticulum, this is a modified endoplasmic reticulum, and more specifically, we know we have smooth versus rough ER, so this is smooth ER. It's modified smooth ER. What's its function here? This is going to store calcium, which is important for muscle contraction. That's Ca2+, that's a calcium ion. I'll abbreviate calcium like that a lot. Excuse me. Let me draw a picture of a myofibril. So here's this myofibril made up of these different sarcomeres, like this. And I'll do the sarcoplasmic reticulum as purple. So let's say that this is my sarcolemma, that outer boundary, that plasma membrane. Within that, in this sarcoplasm, I have these myofibrils, and they're surrounded in this sarcoplasmic reticulum. So it just goes longitudinally like that. And then at certain points, we have these intersections of sarcoplasmic reticulum that are running perpendicularly. So I'll just scatter them throughout. And these intersections come in pairs. We have something in between those pairs. So let's identify what we have. Let's stick with the purple since we're still on sarcoplasmic reticulum. So sarcoplasmic reticulum is storing this calcium, and these perpendicular runnings, we'll abbreviate them TC for now. This is terminal cisternae. So a cistern is a tunnel. I think they're underneath France or Italy, something. There's like a tunnel system. They call them the cisterns. So cisternae. And then terminal is your end point. So as you're going along the sarcoplasmic reticulum, you end at a terminal cisternae there. And then in between those are something called a T-tubule. So T-tubule. And all this is an extension, an inward projection of our sarcolemma. So you can see up top, you can see the sarcolemma, and you can see it indents like this. So it makes this hollow tube in there. Okay. So that's our T-tubule. I want to see if there's anything else we need. I think that's it for now. Oh, and then these things, if you combine a two terminal cisternae and a T-tubule in the middle, that's called a triad. So when we get into the physiology behind all these things, we want to understand the words that we're using. So these are all new words. Myofiber, it's a single muscle cell. It's modified plasma membranes called a sarcolemma. The sarcoplasm, the cytoplasm of the muscle cell, it has these cytoplasmic inclusions, myoglobin, which is this pigment that holds oxygen. Muscle is storing its own oxygen for muscle contraction. We'll talk about that later, and we'll see why glucose is important. It's how we get our ATP. We'll talk about why ATP is important as well. The nuclei, we know that these are multinucleate from the fusion of cells during development, and now we're going into these other parts of cells like this modified cytoskeleton called the myofibril. There's hundreds or thousands of these in one muscle cell. And it's surrounded by this modification of the smooth endoplasmic reticulum, and that's called the sarcoplasmic reticulum. We'll get storage of calcium there, and at certain points of the sarcoplasmic reticulum, we get this perpendicular running, and those are called terminal cisternae. They come in these pairs with something in the middle called a T-tubule, which is an inward projection of the sarcolemma.